Hello everyone, my name is Giulio Aceto and I recently finished my PhD in Silong Wang Lab at the McGill University of Montreal, where I studied the small cell carcinoma of the ovary hypercalcemic type, also known as SCOHT. SCOHT is a very rare cancer that develops in the ovaries of young women. The average diagnosis is at 22 years of age, and it is caused by one mutation in the SMARCA4 gene. SMARCA4 contributes to the expression of many genes that are important for development through its chromatin remodeling activity. In other terms, SMARCA4 helps opening up the DNA to help the expression of specific genes that are important for the normal cell to function and the body to develop. Although we know a great deal about how SMARCA4 functions, much is still unknown on how the mutation of SMARCA4 can specifically cause this rare disease that is SCOHT. And this is exactly what we wanted to understand during my PhD. And specifically, we asked the question of how the SMARCA4 mutation can lead to the development of this type of cancer. To answer this question, we examined the effects of SMARCA4 on the organization of the DNA of the chromatin and on the expression of specific genes that are important to promote SCOHT. This study was published in the scientific journal Nature Communication, and I am particularly proud today to share with you these results. Before looking at the results though, there is one important concept that needs to be introduced: oncogenes and tumour suppressors. Oncogenes and tumour suppressors are the basis of the formation of many cancers, and they are genes that can be expressed by our body. Although oncogenes in normal cells, they are usually not expressed, or they are inactive if you want, while the tumour suppressor genes, they would be present and they would be actively suppressing the formation of tumors, especially by preventing oncogenes to be active to perform their oncogenic role. To use a more practical analogy, you can imagine that an oncogene is like a foot pushing on the accelerator of the cellular growth. It promotes the tumor growth, while the tumour suppressor is like a foot put on the brake that will prevent the cell from growing, that will prevent the oncogenes from accelerating the cellular growth. The case of SCOHT is both remarkable and mysterious. The loss of the tumour suppressor SMARCA4 is essentially the only genetic event that is known to cause the malignancy, that is known to cause SCOHT. But then, in theory, if you lose the tumour suppressor SMARCA4, you can imagine that there will be other tumour suppressors that are able to stop the cells from proliferating. And in particular, SCOHT cells, they retain one of the most important tumour suppressors that we know, p53. In theory, p53 should be able to suppress the growth of those cells that are potentially dangerous, that have lost SMARCA4 and they are ready to form SCOHT. Therefore, to really understand how the loss of SMARCA4 can cause SCOHT, first we need to understand how the loss of SMARCA4 can subvert this tumor suppressive activity of p53. And this brings us to our discovery. In our study, we found that the loss of SMARCA4 redistributes a protein called p300 across the chromatin, which is the structure that packages the DNA in the cell. p300 is an important protein because it allows to add a chemical signal to certain proteins called histones that help storing the DNA, help organize the DNA. This chemical signal, called acetylation, function very simply by allowing the expression of specific genes. So what we observe is that when SMARCA4 is lost, p300 relocates near those genes that are important for the tumor progression. And by adding this chemical signal, this acetylation, p300 is able to express those specific genes, mediating the tumor progression and the cell proliferation. In other words, when SMARCA4 is lost, p300 generates an oncogenic signal. is like that foot pressed on the accelerator of tumor growth. And we also found that this oncogenic signal that is important for the expression of these genes can overcome the ability of the tumor suppressor p53 to repress these very same genes. Therefore, we can say that the loss of SMARCA4 has a dual effect. On one hand, it directs p300 towards genes that promote tumor growth. On the other hand, this redistribution of p300 overrides the tumor suppressive potential of p53. This altered balance caused by SMARCA4 deficiency, by SMARCA4 mutation, might help us understand how SCOT can be such an aggressive disease even if it retains the most important tumor suppressor ever discovered, p53. Identifying this molecular mechanism also allowed us to identify new targeted therapies that could benefit SCOT patients. We use an experimental compound, A485, which inhibits the acetyltransferase activity of p300. Using this compound, we were able to suppress SCOT growth in both cell-based and animal-based tumor models. In our studies, in the models that we tested, A485 also didn't show any sign of toxicity or adverse effects. However, it is important to emphasize that these results are just preclinical. Further studies will be required in the future to establish if this potential treatment, is this experimental molecule A485, could be used for the treatment of SCOT in patients. Nevertheless, our work identifies p300 as a potential vulnerability of SCOT and also identifies a potential mechanism by which SMARCA4 loss can cause these tumors even in presence of the tumor suppressor p53. For me, this publication represents many years of work, but above all, it represents a starting point. These discoveries will allow us to ask new questions and help us to better understand SCOT, which will allow us in the future to discover new targeted therapies that could benefit SCOT patients. But none of this would have been possible without the families, the patient associations, and all of those that support the research on rare disease and rare cancers. And I would especially like to thank Emma Per Sempre and the Small Cell Ovarian Cancer Foundation for supporting our research throughout these years. Without you, all of this wouldn't have been possible. I find it particularly inspiring that the memory of Emma, of Stephanie, and so many other young women that have been affected by this terrible disease can bring together families, scientists, doctors, together with a common objective, trying to defeat and once and for all eradicate SCOT. The road ahead is still long, but today we can say that we have taken an important step towards the right direction. And with this, I would like to thank you all for believing in our research and for keep working with us to try to defeat this terrible disease. And finally, I would like to announce a personal challenge with which I would like to support the research on this disease. On September 21st, I will undertake an Everesting challenge, in which, in one single bicycle activity, I will attempt to climb the height of Mount Everest, 8,848 meters. Every climb and every effort will be dedicated to Emma, to Stephanie, and all the patients affected by SCOT. The goal will be to raise funds for Emma Per Sempre and the Small Cell Ovarian Cancer Foundation to make a concrete contribution for SCOT research. Thank you again, everyone, and I'll see you in September.